Thanks to the organizer giving the opportunity to show our new function of AROD1A in Ewing sarcoma. So I want to give a brief introduction about the Ewing sarcoma. So Ewing sarcoma is a highly aggressive cancer, bone and soft tissue cancer. So this is mainly formed the chromosomal, thank you, chromosomal translocation between the 22 and 11. So basically the N-terminal, basically the N-terminal portion of the EWS part is fused with the C-terminal portion of the Fly1 to forming a EWS Fly1 oncogenic protein. So that's the one, it's aberrant transcription factor which drives the oncogenesis. So the majority of the patients, like 90% of the Ewing sarcoma patient has this fusion gene, which is more characteristics. So today I'm going to talk about briefly how we identified the novel interacting partners of the EWS Fly1, and then I'm going to show the EWS Fly1 splicing function, and then I'm going to show today about the new function we discovered, the EWS Fly1 as a chromatin regulator through the BRAF complex. So I apologize for the complicated picture. So the take-home message here is this is one of the proteome analysis. So we found that the EWS Fly1 associated with the many splicing factors and then involving in splicing. So I want to point it out that the EWS Fly1 direct binding partners PRPFX, PRPF6 and DDX5, DHX9 and HNRNPK, and then it will access the entire spliceosome complex and involving in alternative splicing. So today I want to focus on the BAF complex. Both Jeff mentioned that the BAF is also known as P-SNF complex. So we also identified the EWS Fly1 directly associated with the several members of the BAF complex. So the BAF is, as they already mentioned, the P-SNF, it's a chromatin remodeling complex. It's to play an essential role in differentiation, proliferation, and DNA repair. So BAF complex is mainly described as a tumor suppressor or oncogene, depending on which subunit is mutated. So this is the entire BAF complex, which consists of many subunits, which is essential for the entire activity. So it has several core subunits, and then the main ATPase subunit coming from the BERG or BERM, which is mutually exclusive in a given time point, like either BERM or BERG is associated with the entire BAF complex. So and then I want to focus on the AROD1A, which is the center sac fold of the BAF complex, which is also known as rich interactive domain 1A, which is also known as BAF 250. So as Jeff mentioned so these BAF complex proteins are often called as different names, like BAF or SMARC. So today talk I'm going to stick with the BAF. So next I want to show how the EWS Fly1 associated with the spliceosome complex, and then it's involving in splicing function. And then I'm using AROD1A as an example. So the AROD1A is existing in two canonical isoforms, the long isoform and then short isoform, which lacks the N-terminal portion of the exon 18. So for the validation purpose, what we did is we designed the primers in the exon 17 and then end of the exon 18 in order to see the two product. So these primers product will give the long and short isoforms. So this is the human mesenchymal stem cells, one of the progenitor of the Ewing sarcoma, which shows that the control HMSC cells without EWS Fly1 showing a lot of short isoform. That's a control. And then when we put the EWS Fly1 into the HMSC cells, it switches to the long isoform. So this is the canonical TC32 Ewing sarcoma cell line, which shows that the wild type containing the EWS Fly1 has more long isoform. When we remove the EWS Fly1 from the Ewing sarcoma cells, it's converted into the more short isoforms. Next, we also shown that the protein, when we knock down the EWS Fly1, the AROD1A long isoform protein levels also going down. So we also validated with the 21 Ewing sarcoma patients, and then all the patients are consistently expressing the long isoform. So these are the heat maps showing that each individual patient showing the long isoform of the exon 18. So this clearly suggests that the Ewing sarcoma patients have the long isoforms are well expressed. So And then, so the EWS Fly1 is basically drive the ARID1A alternative splicing, and then it's preferentially prefer the long isoform. And then this also validated in different Ewing sarcoma cell lines. So next I want to introduce about our lead compound YK4279. The analog of the YK4279, TK216, it's currently in the clinical trial. So we tested our lead compound, the YK4279. The cells were treated with 3 micromolar of YK4279, and then we tested in the different time points, and then to see if there is any ARID1A isoform switch. So we do see after 8 hours of treatment, like it's completely switching from the long isoform to the short isoform. So this is kind of encouraging, like how the drug is acting, modulating the splicing. So next we want to show that if the YK4279 is highly drug specificity, the target specificity, and then we use a doxorubicin as a control. When the cells are treated with the doxorubicin, we didn't see any changes. There is no isoform switch. And then also we tested our YK4279 in the control HMSC cells. We didn't see any changes. We do see a lot of so this clearly indicates that the YK4279 is highly target specific, and then it should require the EWS Fly1 for the mechanism. So next, we also, in collaboration with Mona Batish, we showed that the YK4279 switching the long isoform to the short isoform. So the way we designed the experiment is like we put the red probes in the exon 18 region, the N-terminal portion of the exon 18, which is unique to the long isoform. And then we put the series of green probes on the common region between the L and long and short isoform on the exon 20. So we expect, like if the cell's expressing the long isoform, then it will show both red and green together to form a yellow color. And then if the cells show expressing on the only short isoform, it shows the green color. So this is the overlay picture, which shows that the untreated has a lot of yellow cells. So that means like the wild type cells contain a lot of long isoform. And then sometimes we do see a red one. That's a technical issues. But majority of the the S enantiomer treated YK4279 switches the long isoform to the short isoform. So this is the graphical representation, shows that the YK4279 treated cells shows more long isoforms. We also tested with a series of normal cells with the lymph, ovary, thymus, tonsil, and uterus. We show that the short isoform is expressed more. So this clearly indicates that the long isoform is highly specific, like it's expressed only in the cancer condition, especially in Ewing sarcoma. So next we want to pursue the functional aspects of why the ARID1A is important in Ewing sarcoma. So for that, in order to answer that question, so I created a series of construct which endogenously I removed both isoforms of the long and short isoform, the ARID1A, and then reintroduce each isoform individually, the long isoform and short isoform. So this is one of the figure, the Excel legend measuring the growth, showing that when we remove the ARID1A, both isoforms, the cells are significantly stopped growing, like it's growing very slow. And then when reintroduce the long isoform alone, it can able to recapitulate the growth for more than 50%. And when you introduce the short isoform, it further decreases the cells. So next, we also want to test the oncogenicity, and then we did the soft agar colony formation assay to show that the colony formation number, when we reduce the ARID1A, both isoforms, we do see a very few colonies. When reintroduce the long isoform, we do see a lot of colonies, number of colonies, but this was not recorded by the short isoform. So next, further, we also want to show that isoform-specific function. So what will happen, like if there is any rescue, if you reintroduce the long isoform alone. So for this purpose, I created the stable long isoform overexpression construct. From that, I removed the EWS Fly1, which is the oncogene driver, so to make sure that still it can able to survive or still it can able to grow. So as we know that when we remove the EWS Fly1, the cells won't grow, so that's why the curve is here. And then when we remove the EWS Fly1 from the ARID1A long isoform overexpression, we can able to recover the growth significantly, like more than 50%, and then the growth is further growing. So we do consider the excision experiments up to 72 hours, because after that, whatever the cells will become oversaturated the cells. So next, surprisingly, we saw that when we overexpress the short isoform, so it's completely kills the cells, even though the EWS-FLI1, the wild-type EWS-FLI1 is still present, but it's completely killed the cells within 3 days of when we overexpress the short isoform. So this clearly indicates that ARID1A long isoform has a significant contribution to the Ewing sarcoma oncogenic growth. So next, I want to talk about our new discovery of EWS-FLI1 as a chromatin regulator through the BAF complex. So this, I already shown this network before. So first, we want to try to validate this network is actually happening inside the cells. So what we did is we did a immunoprecipitation using the FLI1 antibody to pull down the EWS-FLI1 and then to see whether it can able to associate it, pull down all the BAF complexes. So it shows that it can able to pull down all the BAF proteins except the ARID1B. So the ARID1A and ARID1B are mutually exclusive proteins. So at a given time point, either ARID1A or ARID1B are associated with the BAF complex. So this is clearly indicates that our EWS-FLI1 associated with the BAF complex require ARID1A. So next, we want to pursue some more functional studies, like whether the EWS-FLI1 is directly associated with the ARID1A. So what we did is so I synthesized the protein, the fragment protein using the in vitro transcription translation reaction to produce the long fragment and short fragment. Basically, I used this portion to synthesize completely the long version and short version, which lacks the N-terminal portion of the exon 18. And then we did the pull-down experiments using these ARID fragment coupled in the beads. And then I used the TC32 nuclear lysate pass-through and then to see if there is any protein interactions. So this clearly shows that the EWS-FLI1 can able to bind to the long isoform, not the short isoform. So next, we want to test whether this interaction is direct or indirect. So what I did, I set up the ELISA experiment to coat the long and short fragments into the bottom of the well, and then I put the recombinant protein of the EWS-FLI1. And then it clearly shows that the EWS-FLI1 can able to bind only the long fragment, not with the short isoforms. Also, we showed that the disruption of EWS-FLI1 and ARID1A interaction by YK4-17 in a dose-dependent manner. So we do see a gradual decrease when we increases the dosage. So this clearly indicates that the YK4-17 can able to dissociate the interaction between the EWS-FLI1 and full-length ARID1A protein. So next, we want to see the functional readout. So I used here the ATPases as the functional readout to measure the complex activity. So for this, we created the construct, the wild type, and then remove the knockdown of ARID1A, both isoforms into the cells. And then I measured the total ATPases activity. I didn't see much difference in the total ATPases activity. But then I did the pull down with the EWS-FLI1 to look at the specific EWS-FLI1 associated complex. And then we do see a significant decrease of when we knock down the ARID1A. So that clearly indicates that there is a significant loss of ATPases activity when the ARID1A is removed. So the reason is when the ARID1A was removed from the complex, so the EWS-FLI1 is not associated anymore with the BAF complex. So it completely loses the connections between the EWS-FLI1 and BAF complex association. But still, we do see some ATPase activities are coming. So that's because we already know that the EWS-FLI1 is associated with some other the known interacting partners of RHA or DDX5. So that may contribute some ATPases activity. So that's why still we do see some ATPases activity signal, but it significantly dropped when we removed the ARID1A. So with that, I'm going to summarize with our model. This is the current working model. So I showed that the EWS-FLI1 can physically associate with the spliceosome complex, and then it's preferentially prefer the long isoform for their oncogenic activity and growth. And then once the EWS-FLI1 directly binds to the ARID1A long isoform, and then it can able to access the BAF complex, and then thereby it controls a lot of gene activity turn on and off with the transcription. So when the ARUD1A was removed, so it could not able to associate with the BAF complex anymore, and then it seems to be the cell's going to die. So this concludes that the ARUD1A and the interaction with the EWS-FLI1 can be considered as a potential therapeutic target. So with that, I would like to acknowledge all the members of the Toresky and Uren lab, especially Garrett and Alex. And then also, I would like to thank all our collaborators, Sukover and Eleni from the Vienna, and then Mark and Mona Batish from the Rochester University, and then Brian and Kim from Boston. And then finally, I would like to thank the CCF for our funding. So, and thanks for listening.
Can you hear me?
Yeah.
Okay. So I recall a few years back there was a flurry of publications, and it seemed like every couple of weeks or months another group was reporting their findings that ARID1A was lost in some new tumor type. So I'm guessing from your findings that means that ARID1A is not lost in Ewing sarcoma and seems required for EWS-FLI1 activity. Is that actually true, and do we know that from genomic studies?
Right. So I didn't have enough time to show all the mutation data. So we couldn't find any single ARID1A mutation in Ewing sarcoma patients. Also, like the ARID1A is a well bona fide tumor suppressor. So it's already established in several cancer conditions because of the mutations that they found a lot of mutations in various regions. But since we couldn't find any single mutation, and then the function is completely different in Ewing sarcoma. So in Ewing sarcoma, it's acts as a complete oncogene. It's not a tumor suppressor anymore. So that's why we got excited in the initial, and then we pursued a lot of mechanistic studies to prove that. So the ARID1A is required. So even when we removed the ARID1A, the cells are not surviving. So even though in the presence of EWS-FLI1, so that means the EWS-FLI1 oncogene is required the ARID1A for the oncogenesis as well as the growth. So it's completely new function.
Thanks. Again, Brian from Johns Hopkins. So the isoforms of ARID1A, the long and short, so I didn't quite follow maybe, but does the EWS-FLI, does it control the splicing of that?
Yes.
Or do you feel like just the long form is maybe stabilized because it binds to the EWS-FLI? I mean, so it's kind of almost like it's selected to last longer? Do you feel like there's actual control in the isoform splicing?
Yes. So the ARID1A is mainly regulating the alternative splicing of EWS-FLI1 is mainly regulating the alternative splicing of ARID1A, and it's preferentially prefer the long isoform. So, and then we also did some MG132 experiment, the proteasomal degradation experiment, to show that when we remove the EWS-FLI1, we can able to stabilize the ARID1A. So that means when we remove the EWS-FLI1, so it affects the ARID1A isoform level as well. So this clearly suggests that both ARID1A long isoform and EWS-FLI1 is required for the Ewing sarcoma oncogenicity.